सारा ग्रुप बाहर आ जाए. Your time starts now and Maria is keeping the time. 20 मिनट से मारिया ज़्यादा तो नहीं होगा और उससे कम हो जाए तो we are grateful. चलें जी, go ahead.
तो ओपन कर दो.
It's up to you to organize yourself.
मैम स्लाइड्स ओपन करो.
ये प्रेजेंटेशन ग्रुप 1 की प्रेजेंटेशन कहाँ है?
IBC
तो यहाँ पे तो है ही नहीं प्रेजेंटेशन. ग्रुप 2
IBC सर जो
Can you speak from here?
चलो
Go ahead. बेटा आपका टाइम शुरू हो चुका है.
Hello, Assalamualaikum. Excuse me. Hello, Assalamualaikum. I'm Soma. Today I'm presenting the Group 1 members, and today I'm presenting the topic which is institutional biosafety community. Biosafety community. These are all the members of the my group. And what is the mission of the biosafety committee? This is the first topic and the introduction. Institutional biosafety committee is basically mainly responsible for the safety of the student, staff, and all the working members which are present in the institute. They are mainly they are the safe basically. And the secondly, this committee ensures that all the research conducted in the institute is safe or secure. The persons which are working in the institute are safe. And the thirdly, it works on the different strategies and plans to mitigate the bio risk. That's all from my side, and the second member come.
Okay, assalamu alaikum. As you know, Soma introduced our group as well as our group presentation topic. So I want to continue just the introduction of IBC. IBC is a panel or is a group of expertise that assess the risk associated with research that involve the biological hazard like gene therapy and something like that. If someone doing research or someone doing a project in the lab and they are associated some biological hazards, so IBC is responsible for that to reviewing, approving, and overseeing that the project is safe and they can be conducted inside the lab. For example, a research doing DNA therapy inside the lab, so that may be a problematic because some big problem or some small problem. So they need, first of all, they need to approve his project from IBC, and IBC ensure that this project is okay and there is no critical harms. So this is the main introduction of IBC. And now I want to call my another colleagues to continue the presentation.
After my friend was talking about the introduction, so my turn is to talk about the origins and the evaluation of IBC. So firstly, it was started in 1975. The Silomer Pacific Grove, California, firstly it was started, and actually in order for the projects or rather for the experiments to be conducted in a safer way, so this a kind of a meeting or kind of a conference was conducted. That was in 1975. So because of which some responsibilities, some rules and regulations were made, and also some investigators were made aware about the rules and regulations. So the rules and regulations were made according to the NIH guidelines, which were in 1976 and 78. And so the rules were actually made to, in every institute and organization should be made for the experiments to be conducted in a safer way. So that was named as IBC. After that, and some terms were used, for example, biohazard was used, and biosafety, that was changed to biosafety, and also trainings and special surveillance and observations were done also on this. So after that, the IBC, actually I'm talking about the composition of the IBC. It is consisted of mainly at least 5 members. More members can be also included, but firstly, however, at least 5 members should be there. Firstly, 2 members are selected from the community, and secondly, 1 member is actually selected or rather from expertise in animal containment or rather zoologist, and another from plant, that is botanist, and another person that is selected, that is biological safety officer. So the next turn will be presented by.
Now I talk about the mission of the IBC committee. The first one is the standard, to establish standard and procedure for the risk management. Because for the biosafety and biosecurity of one health. And the second one is insurance of genetic manipulation.
Continue.
The second one is
Continue.
insurance of genetic manipulation. In this IBC established foreign linkage with foreign biosafety committees and relative agencies to ensure that the genetic manipulation practices in Pakistan or in any institute absorb international biosafety measures or concern universal code of conduct. And the second one is educate public on biosafety issues. Because the mission of IBC is not complete if they will not educate our public about the biosafety, these biosafety issues, biosafety problems, and their management. This is my turn. Continue by
I'm going to discuss more about the missions of IBS. First one is to facilitate appropriate experimental conditions to provide safe and secure research in the laboratory. Second one is to ensure compliances of policies so that we can avoid the exposure of laboratory personnel, the community, or the environment to these biological toxins or biological modified organisms. Third one is the recommendations required for import and export, as they have to consider the application of import or export of these biological toxins on recommendations with TAC, basically that is Technical Advisory Committee, which allows or reject the commercial release of these biological modified organisms in accordance with the biosafety guidelines. That's all from my side.
Hello, my name is Muhammad Shafiq and I am here to summarize the missions of IBC. IBC is responsible for reviewing, approving and monitoring all the research projects which involve biological material and pose some level of risk to plants, animals and humans. So the goal of the committee is to ensure that work is being done in a very safe manner. The committee's main purpose is to perform critical hazard risk assessment, provide guidance and create awareness about the safe handling of various biohazardous materials. It ensures the compliance of policies in the light of institutional, national and international recommendations.
Next step. Okay. Next step is the opportunities and challenges of the IBC. And the first one is the institutional support. And here's the awareness related opportunities for IBC. And for awareness, institutes conduct seminars, workshops, and trainings can be arranged for the awareness about biohazards of the laboratories, risk management issues. And the next one is the code of practices for the safety in the microbiology and biomedical laboratories, like the appropriate handling of the equipments and the appropriate handling of the hazardous materials, like the biological, radiological, recombinant DNA toxins, extra. And the next one is the workspace in implementation of decisions to facilitate IBC. Regarding making decisions in the rejection, implementation, and the exemptions of exemptions for the conduct of research studies in the laboratories. Thank you.
Adil here, and in my section I will discuss some opportunities and challenges. One of the opportunities is the public support, that ये जो IBC है इसमें अपॉर्चुनिटी एक हो सकती है कि पब्लिक को इन्वॉल्व किया जाए अपनी कामों के लिए, दूसरा है कि गवर्नमेंट से सपोर्ट ली जाए, different services के लिए like policy, police service, waste management service and others. इसके अलावा ये कि जो कमेटी है उसको एक प्रॉपर साइंटिफिक और गवर्नमेंट एजेंसीज जो साइंटिफिक बॉडीज होती हैं उसकी गाइडलाइंस के हिसाब से बनना चाहिए, लाइक CDC हो गया, सेंटर फॉर डिजीज एंड कंट्रोल प्रिवेंशन, इसके अलावा NIH, नेशनल इंस्टीट्यूट ऑफ हेल्थ. फाइनली कंफ्लिक्ट ऑफ इंटरेस्ट अगर बिटवीन अमंग द मेंबर्स ऑफ द IBC हो तो उसको डिस्कस करें और पॉसिबल सोल्यूशन सजेस्ट कर दें. दैट्स इट. अदर पोर्शन विल बी डिस्कस्ड बाय मरियम.
Here's Mariam, and I will discuss about the registration of all the research and training about IBC members. First of all, registration of all the research conducted in the institute is necessary. There should be a proper system to conduct registration of all the research because it can provide an audit point for the later accountability. And as for concern, training practices should be done because there are two types of training sessions. One will we discuss about general laboratory practices, in which we discuss, we will teach them and educate, train them how to use personal protection equipment. And second, proper use of chemicals, hazards related to them, what we should do in emergency responses, what classifications of organisms are used and what hazards are attached to them, and what we should do in the later. And the training sections and workshops should be done twice annually. And all the IBC members and students must know how the safety should be used in such emergency situations and other hazards.
We usually protect ourselves, but as a researcher, it's our responsibility to make ensure the safety of others, including animals and environment, as we are one health. It is very important to dispose the waste material from labs properly. For the proper waste disposal, first of all, we must train our researchers, lab staff, and especially the housekeeping staff about the different type of waste and their proper disposal and segregation. There must be different type of containers for different type of wastes, like waste containing blood, the sharp equipments, and that's it.
I would like to explain the role of IBC in collaboration with IACUC, that is Institutional Animal Care and Use Committee for Animal Research. These type of research which involve the use of transgenic or cloned animals, use of recombinant DNA molecules in animals, and transfer of human gene in animals must be registered, reviewed, and approved by IBC and IACUC before the work is initiated. The role of IBC and IACUC is well defined. That is, IBC will review the risk to human health and environmental escape in the wild breed, whereas IACUC review for the animal welfare and risk to other animals in the facility for the spread of the vectors. Thank you.
Okay, continue. Dual use of research, we know that we can use, people can use research as for beneficial purpose, or it could be harmful for things like we can use it as pharmaceutical industries or for vaccination, diagnostic, etc. Or it can be used for somewhat harmful purposes. Second thing is containment of biohazards. Containment is the term used to describe basically methods, practices, procedures, facilities, and equipment used to safely manage biohazards material in the laboratory. Basically, the purpose of containment is to reduce or eliminate exposure to people or the environment to potentially hazardous agents.
Okay, so IBC has a very difficult challenge, like it is to minimize the bioterrorism by studying a project with every aspect and from every angle, so as to reduce the dual use of any information which is contained in a project or a thesis. So there are some very important opportunities for the IBC, and they can gain information from everywhere. Like we can organize the meetings of one IBC of an institute with the other IBCs of the other institutes, so as to gather information and as to get some information from the experiences of the other IBCs of the institutes. And we can also conduct the meetings of the IBCs within an institute, like with the other committees of an institute with the IBC, like with the committee which deals with the zoology, or a committee which deals with the botany, so as to gather the information which is like everywhere in the institutes, and so as to apply the IBC rules effectively and efficiently. Thank you.
Faiza Hadi is here. I'm PhD student here at ICCBS, and I basically talk about the key points on which institutional biosafety should work. These points are based on 5 W's: who, what, where, when, and why. And the main points which should be evaluated by IBC committee members are: they should evaluate the expertise of principal and investigator and staff to conduct the work. They should examine the experimental protocol that are submitted with grant application. They should evaluate the potential dangers of the work that has been done, that will be done, sorry. Evaluate the biological containment plans and facilitate as per NIH guidelines. Thank you.
Assalamu alaikum. My name is Sayyid Ritza Ali. IBC core facilities are designed to merge technologies, provide sharing of research tools and resources. These facilities are accessible to all investigators at the institute. Some facilities are accessible to researchers outside the university or institute at fee levels, from free or no costing to full costing. Facilities that should require IBC review are researches that involve bacteria, toxins, human blood, tissue, or cell lines, generation of transgenic plants or animals, viral containing materials. IBC core facilities should focus on the following areas: training of core personnel. Staff are fully informed on type of sample and hazards associated with those samples. Biosafety inspection and facilities annually or half yearly, database management, and upgrade of new records. Thank you.
This is Junaidullah from HEJ. I will discuss the accountabilities that are related to the BSL level 2 and 3. If we define the biosafety level, so the biosafety level isolate the dangerous biochemical agents such as bacteria and viruses from the outside of the environments. If the committee members divide the committee into the subcommittee members, so one of the committee member should periodically perform safety inspection of the labs which are involved in the biological research that require the biosafety level 2 or 3. The committee members are also accountable to investigate the accidents that sometimes happen in the lab due to some infectious agents. The IBC shall also answerable for ensuring institutional compliance with the applicable guidelines of the state and the government. In addition, the IBC is also accountable to monitor the research involving the harmful agents. Again, these may be some harmful nucleic acids or bacterial or some other infectious viruses. So the committee should ensure that all these steps should be followed in the institutions where the BSL 2 or 3 lab facility are available. Thank you.
Assalamu alaikum, this is Varda from BCMD. I'm going to present here the biosafety levels. Biosafety levels are the laboratory that are designed to control these pathogenic microorganisms. For example, you are a microbiologist and you are working with a harmful organism. ठीक है, अब आप किसी हार्मफुल ऑर्गेनिज्म के साथ काम कर रहे हो तो आपके जहन में एक चीज़ आती है कि ठीक है ये हार्मफुल है तो मुझे इसके लिए अगेंस्ट जो है प्रोटेक्शन चाहिए। मैं काम कहाँ करूँगी, मैं कौन से सेफ्टी इक्विपमेंट्स यूज़ करूँगी जिनको यूज़ करते हुए फिर मैं इनके ऊपर वर्क कर सकूँ, मैं इनको स्टडी कर सकूँ। तो बायोसेफ्टी लेवल्स जो हैं बेसिकली ये वो लेवल्स हैं, या ये हम कह सकते हैं कि वो लेबॉर्ट्रीज हैं जो कि हम डिज़ाइन करते हैं अकॉर्डिंग टू आवर रिक्वायरमेंट, अकॉर्डिंग टू द ऑर्गेनिज्म वी आर डीलिंग विद। तो सबसे पहले बात कर लेते हैं बायोसेफ्टी लेवल 1 माइक्रो ऑर्गेनिज्म एंड 2 की। दोनों को चारों को एक साथ लेकर चलूँगी। तो 1 और 2 की बात की जाए तो 1 में आपके पास जो माइक्रोब्स आप डील कर रहे हो ये ऐसे ऑर्गेनिजम्स हैं जो कि हेल्दी पर्सन को कोई नुकसान या कोई इन्फेक्शन कॉज नहीं कर सकते, जबकि बायोसेफ्टी 2 में जो हम ऑर्गेनिजम्स यूज़ करते हैं वो मॉडरेटली हेजर्डियस हो सकते हैं, कुछ हद तक हो सकते हैं कि अगर आप बीमार पड़ जाते तो कॉज योर डिजीज़। ठीक है, इसके अलावा बायोसेफ्टी लेवल में कुछ इंडिजीनियस ऑर्गेनिजम्स भी आ जाते हैं, यानी कि वो ऑर्गेनिजम्स जो कि आपके बॉडी पे ऑलरेडी प्रेजेंट होते हैं ड्यू टू अगर उनको फेवरेबल कंडीशंस मिल जाती है, आपका इम्यून सिस्टम जो है वीक पड़ जाता है तो दे कैन कॉज यू डिजीजेस। ठीक है, तो बायोसेफ्टी लेवल में आपके पास नॉन पैथोजेनिक इकोलाई आ जाता है, मतलब नॉन पैथोजेनिक इतना ज़्यादा डिजीजेस कॉज नहीं करेगा। और 2 की बात की जाए तो स्टेफिलोकोकस ऑरियस, क्योंकि ये ऑर्गेनिज्म डिफरेंट सरफेसेस आपकी स्किन पर प्रेजेंट होता है तो इट कैन कॉज यू हार्म। 3 और 4 की बात की जाए तो यहाँ पर जो है मोस्ट पैथोजेनिक ऑर्गेनिजम्स आते हैं। ये मोस्टली वो ऑर्गेनिजम्स होते हैं जो कि इजीली एरोसोल्स के थ्रू या फिजिकल कांटेक्ट के थ्रू जो है ट्रांसमिट हो सकते हैं। तो 3 में और 4 में दोनों में, 4 में अगर बात की जाए तो 4 में तो मोस्ट डेंजरस हार्मफुल टाइप ऑफ ऑर्गेनिजम्स आ जाते हैं। तो ये वो ऑर्गेनिज्म हैं जो कि डिजीज़ तो कॉज कर सकते हैं, बट वो डिजीज़ जो है इट कैन बी लीथल, इट कैन बी अच्छा जी। इसके अलावा लेवल 3 की बात की जाए तो इसमें जो है अच्छा लेवल 3 की बात की जाए तो इसमें जो है वो ऑर्गेनिजम्स आते हैं जो कि इजीली रेस्पिरेटरी सिस्टम के थ्रू ट्रांसमिट हो सकते हैं। तो माइकोबैक्टीरियम ट्यूबरक्लोसिस आ जाएगा। और लेवल 4 की बात की जाए तो इसमें हाईली इन्फेक्शियस ऑर्गेनिज्म दैट कैन कॉज फेटल डिजीजेस और जिनके खिलाफ कोई ट्रीटमेंट या वैक्सीन नहीं है। तो इसमें आपके पास इबोला एंड मर्डबर्ग वायरस आ सकते हैं।
Thank you very much. Thank you. A big hand for it. Excellent. Though we didn't have much of time, but you really performed well. It's really good enough. I mean, with the time that you had, we could have expected this one. The only request that I would have for you is, when I watch the online people, there are some international people also, like from Thailand, there is a girl attending. There are several from outside the country. So we try, I also sometimes mix my Urdu with it, but I didn't realize that when I checked that there are international students attending. So for their sake, I try to speak in English if possible. Number 1. 2. What now I propose is that we talk to these online people for 5 minutes only, and we'll say goodbye to them because we have to use the site for another meeting simultaneously online. So we will say goodbye to them and we continue our group work after 5 minutes the next week. And another activity for you is that the group, if they want, they can photograph together with all of us, group 1, and then next week and next week and next week that we can do an activity. You can have your title page you want, and you can all come, and your Madam Hena is also here, and so is Valia. And if you want, a bearded man can join you. Let's have a photograph with group 1. And online people, there is a hand raised by someone. Namra, you want to say something? We are going to close down and say goodbye to online people. Anybody who wants to say anything online, they can. Now you can see the group on a photograph through. this one also, और है ना फिर मेरे साथ भी शेयर कर लेंगे। जी जी सर। तो आज आप ऑर्गेनाइज हो जाए तो बीच में हम भूल जाएंगे।
सर आप आए ना।
मैं आता हूँ। मैं जरा ये ऑनलाइन वाला पे, हाँ निम्रा कुछ कहना चाहती है? देयर इज अ हैंड, निम्रा जमील, ताकि इनको हम खुदा हाफिज कह दें।
हैंड अटेंडेंस मांग की।
अटेंडेंस मांग की, चलें ठीक है। और ये चैट में कोई चीज़ एनी इम्पोर्टेंट, ये भी अटेंडेंस है, अटेंडेंस है। Okay, all online people, if you are listening me, we say goodbye to all of you. You can just watch our group 1, that they are together. I'm sorry you will miss the rest 3 groups because of reasons that are beyond of our control. So goodbye to online people. We may not be able to talk to you after 10 minutes. You can just watch. Yeah, Aisha kuch कहना चाहती है, attendance है ये भी। हाँ? Attendance है? अच्छा. Just an announcement that online people, those who attended all the session, had a quiz. I have requested Dr. Hina to send you